Hello, and thank you for tuning in to Answers from the Lab, where we share Mayo Clinic knowledge and advancements on the state of testing and science from laboratory leaders and the people who are making it happen behind the scenes. I'm Dr. Bobbi Pritt, Interim Chair of the Department of Laboratory Medicine and Pathology at Mayo Clinic in Rochester, Minnesota. With me today is Dr. Peter Lucas, Vice Chair of Research in the Department of Laboratory Medicine and Pathology at Mayo Clinic. Dr. Lucas joined Mayo Clinic in January, so I have the pleasure of working with him for several months now. We're excited to introduce him to our listeners today and to get to know him and his thoughts on laboratory medicine, pathology, and research. So, Dr. Lucas, thank you so much for being with us here today.
Well, thank you very much, Dr. Pritt. It's really a pleasure to be with you today, and thank you for the invitation to join you on this podcast.
Yeah, that's great. Well, maybe we'll start with what I hope is an easy question. Can you share a little bit about your background and what drew you into the field of pathology?
Sure. Yes. So I've been at a few different institutions over the course of my career in medicine. First, as a MD Ph.D. student in the MSTP program at Vanderbilt, and it was there that I really first became aware of the field of pathology because at that time, and this was a number of years ago, I'll just leave it at that.
You don't have to tell us.
Well, at that time, most medical schools, including Vanderbilt, had a fairly traditional curriculum that was very course or topic based during the first 2 years, meaning that we had discrete courses in anatomy and physiology, biochemistry, pharmacology, and so on. And there wasn't the kind of effort that we see today to kind of integrate all those topics into present material in this very patient-focused way, which is, I think, really the more modern curriculum. But I will say that, at least for me, there was really a benefit in that pathology was a cornerstone course that ran the entire second year of medical school. And so as a student, I really saw pathology as a discrete entity. It was a field, and most practicing academic pathologists at Vanderbilt taught in that course. And so I could get to know them and really understand what they did for their careers. In a way, it was kind of like a clinical clerkship that students take in their third year of medical school and other specialties like medicine or surgery. And so that really deep exposure to pathology as a second year medical student, as a discipline, really just had a huge impact on me. The pathologists at Vanderbilt who ran that course were just incredibly charismatic and included some of the giants of pathology like Robert Collins, who's a huge figure in hematopathology. So while I guess I didn't fully appreciate it at the time, that exposure laid the foundation for me. And so when ultimately I completed all my clinical rotations in other fields, I kept comparing the experience I was having to my memory of the pathology course. And honestly, nothing just ever felt as compelling to me. And so I chose pathology as a residency track, and I've just been, so happy that I did. I think that focusing on pathology being the study of the fundamental biology and mechanisms of disease, and our goal of interrogating disease processes through test development, that just really spoke to me because I was always interested in learning the details of why things work, why things don't work, and pathology was just the perfect career for someone who wanted to also pursue those kinds of questions and to perform research into the basic mechanisms of disease processes. So after Vanderbilt, I trained in pathology at University of Michigan for residency and fellowship, stayed on the faculty there as a surgical pathologist, specializing in breast pathology. And I also launched my research lab, which studies basic signaling mechanisms in cancer. And so in total, I was there for 17 years, and then moved to the University of Pittsburgh, the UPMC hospital system. And that move was inspired by my wife, Linda, who was recruited to be the chief of pediatric hemonc at Children's Hospital of Pittsburgh. And we've always run a joint research lab together. So we came as a package deal. And at Pittsburgh, I took the opportunity to pivot just a little bit with my clinical role, and I joined the Division of Molecular Genomic Pathology because at that time they were one of the earliest institutions to establish a surgical pathology focused molecular division. So that meant I could leverage my background in surgical pathology to develop skills and expertise in genomics, next-gen sequencing of solid tumors, and related techniques. And so I really loved that role, and I'm continuing in that role here at Mayo. So as you mentioned, my wife and I just joined Mayo a few months ago, and after 11 years at Pittsburgh, and I'm assuming this role as the Vice Chair for Research in Laboratory Medicine and Pathology, DLMP as we call it. And I just couldn't be more excited. And it's especially fun for me on a personal level because while my career has been elsewhere, to this point, Vanderbilt, Michigan, Pittsburgh, I actually grew up in Rochester, and I went to school here from first grade all the way through high school. So it's just super exciting to come back full circle in this capacity, see Rochester from a totally different perspective, see how Rochester has changed and grown, and it's just really a thrill to have this opportunity to come back. And so I'm just very grateful for this opportunity.
Well, Dr. Lucas, what a great story. And I can't even imagine how much Rochester, Minnesota must have changed over the years. We are just so thrilled to have you as our Vice Chair of Research here in DLMP. And I also love your story about the importance of role models and your pathology during medical school. And I know that a lot of our listeners probably are at academic centers where they've changed the model of medical schools. Even if you don't have your own pathology course, so to speak, anymore, I still think that importance of a role model, it's just so critical. And the studies that they've done show that having good role models that you could look up to and say, wow, that's a pathologist, I want to do what they're doing, is one of the most important reasons people go into pathology. And having spoken to some of your former trainees, I know that they look to you as one of those role models.
Oh, well, that's very kind. But I totally agree with you that having, these role models, you can't overestimate the impact that a single person can have on somebody who's coming up early in their career, and having those people that you look to as representatives of what it might be like to have a career in a particular area, it just has so much impact. And a lot of it's serendipitous, right? Because we sometimes run into people who change our lives just because of who they are, and our chance interactions and opportunities to view those people as mentors. So there's some randomness, I guess you can say, and serendipity in the whole
but you
process.
never know. And we have a whole group of residents and fellows that are excited to work with you now. And that kind of leads me into my next question. Because you're in this new role at Mayo Clinic, can you tell us a little bit about what you'll be working on, particularly focusing on your research, since that's a big part of what you do?
Sure. So yeah, this is a great question. So one of my first objectives in thinking about how to, advance research, as you say, is to first listen first, to make kind of a landscape analysis. I want to just learn with as much granularity as possible what the research is going on currently in DLMP, where we have strengths, where opportunities lie, how what we do fits into the broader scope of research at Mayo. And so I'm doing that. It's kind of like drinking from the fire hose, because every day I stumble onto something that I had no idea was going on, and it's very invigorating and exciting, and just learning that there is some incredibly inspirational work happening here. We've got some amazing investigators. It's just exciting to learn all about this. And along the way, as I'm learning, I'm starting to put together in my mind where we might have opportunity to build out unusual levels of expertise, unparalleled strength. And ultimately, and this is where I need the help from others, and I'm working to think about assembling a task force to help with this. I hope that we could stand up maybe 4 or 5 areas or topics of what I would call special research emphasis, areas where we've got this unique opportunity to really move the needle in research by assembling this unusual expertise, leverage unique biospecimens that Mayo is especially well known for, technologies that we might be unusually positioned to tap into, and establish these areas as sort of pillars for future growth in research and for training our junior faculty and our earlier stage trainees to kind of really make progress in these particular areas. And part of this was inspired by something that our new chair, Eric C., Dr. Eric C., said to me a while ago. He said, you can't boil the ocean, but you can boil the heck out of some pretty big pots. And so
I like that.
I do too. It really resonated with me, and I really embraced that concept, meaning that we can't do everything, but if we choose wisely, we can do some things really well. So I think we can make the biggest contributions to biomedical research, ultimately, leading to improved diagnostics and care of patients. That's the ultimate goal. So have the biggest impact in this if we come together in teams around a few specific areas. And so my goal is that, ultimately, when everyone everywhere is asked about these particular topics, Mayo DLMP is the place that is top of mind. It's the department that really had or is having the most impact. At the same time, I do want to stress that while I think this is an important goal to identify some areas of, as I say, special research emphasis, that doesn't mean that we would only want to support those working in those particular areas. I recognize that some of the most important discoveries in science have come from those who are working around the edges, exploring new ideas that didn't necessarily fit well with established programs, or were not, the focus per se of a department or a larger program. We've got to support innovation that falls outside those lines and to recognize good work that's being done across a spectrum of areas. So I guess, a final word on your question of what I'll be working on. And that is that another key objective for me is to break down barriers that might exist that limit research progress in whatever areas are being studied. And this can include addressing challenges such as protected time, core services, availability of instrumentation, administrative support, all the things that are necessary to help folks engage in research and make facile progress. It's a big job. It's something I'll be working on with my administrative partner, Linda Sanders, and our whole team. And again, something I need to understand better here, what the limitations are and how we can make things as facile as possible. But ultimately, we can stand up areas of special research emphasis, but we also have to make acting on those areas and that mission as easy as possible.
Well, that's really exciting. I'm really excited to hear your plans and how you're going to support research in maybe some of those big pots, as Dr. C said, but also across the whole spectrum, not leaving anyone out. I don't know, maybe infectious diseases, microbiology will be one of those big pots. We'll see. But well, my next question will lead from this one. The field of lab research has certainly changed over the years. There's an increasing emphasis on metagenomics, molecular testing, new advances in anatomic pathology. So what are some of the challenges that you think laboratory research will have to overcome to continue to make progress?
Yeah, another great question. And it follows on, some of my last comments, the challenges. I think that there's a little bit of a paradox going on right now in laboratory research, and in that fundamentally our capabilities to conduct exciting research, probe mechanisms of disease, they're unparalleled with rapidly developing technology, some of the things we just mentioned, metagenomics and so on, and these technologies, instrumentation, informatics, the advances have just been incredible and super fast. At the same time, I think the paradox is that the accessibility bar, if you will, is being raised higher and higher. And I think there are several different aspects to this. Number one, simply the cost of doing science is increasing at a pace that outstrips traditional grant funding mechanisms. That the NIH budget is just not keeping pace with the cost of what it takes to do cutting edge science. And so I think that means that while it's super important that we continue to aggressively pursue federal funding, NIH funding, and I say that in part because I think the peer review process just makes our science so much better. It's not just about getting the funding, but it's also about increasing the rigor and validation of our scientific goals. So I think it's super important that we do that, but we also have to look elsewhere to make up for what's kind of a growing gap in the real cost of doing science. I think a second challenge is that the expertise needed to then take advantage of all these advances in technology is getting rapidly more and more complex. And so it means that to be successful in making some of these really innovative discoveries and pushing the boundaries, we have to leverage teams more and more. And I think about that, especially when it comes to the informatics side of doing science. Scientific investigation now depends more and more on abilities to handle big data. So all of the omics, genomics, you mentioned metagenomics, transcriptomics, proteomics, metabolomics, you name it. All of these things generate enormous amounts of very complex data. And at least for investigators like me, we need help. And it's not always obvious how to get that help. But I think this is something that institutions like Mayo needs to address as to how to bring together teams of people with distinct areas of special knowledge and then to be able to really drive complex projects forward. Finally, I'll just say that one of the things that is most important as a challenge is time, right? We all just time to sit down in uninterrupted fashion and time to just think. It's really become a commodity in our hyper scheduled worlds with so many different distractions competing for our focus. So, and the solution to that is very personal. Everyone's got a different sort of situation, but at least for myself, I have to take a very active and purposeful series of steps to ensure that I can preserve time in my day to really think at a level that allows me to develop ideas for research that can make a difference and try to pull in all this increasing complexity in what science really means. So those to me are some of the top challenges, just the funding, the complexity and the need for teams and the time to actually pull it all together.
I have to schedule time sometimes for myself just to think or brainstorm or put my thoughts together, take notes, etc.
Right. Yes.
Well, you mentioned some really cool advancements, all the omics, as you mentioned, instrumentation, AI. What are some of the advancements or trends in pathology that excite you the most? And then how do you stay current with all of these?
Yeah, well, these are great questions, Dr. Pritt. So, I think you're right. There's so many different things, but I'll mention one thing that's really on my radar, and I'm clearly a little biased in this because as someone who has a background in surgical pathology, but I feel that right now we are at this watershed moment in our ability to interrogate microscopic tissue images, microscopic tissue pathology in ways that just nobody dreamed about even just a few years ago. And there are several aspects to this. There are new technologies to allow us to evaluate the transcriptome, for example, at levels that are approaching a single cell level within tissue sections. Companies like NanoString and 10x Genomics have platforms that are allowing us to do this at increasingly precise levels. There are also technologies to evaluate the proteome, if you will, in a very highly multiplexed way using fluorescence-based advances, kind of immunohistochemistry on steroids. Companies like Akoya and other companies have developed technologies to do that. And there are even advances in image analysis that are deriving insights using AI from just regular H&E stains of tissue sections. And what's being done there is just mind-boggling, deriving information, developing algorithms around prediction of therapy response, prognosis, and so on, just from H&E images that we can't fully comprehend just by looking at ourselves. There are features that the machine learning can derive that we're not able to do as pathologists looking through the scope. And to add to that, and this sort of, part of your question was, how do I keep appraised of that? I think part of it is just being present and taking in as many seminars as you can. And this leads to this thing I just learned about a week ago from a seminar that was held by our Advanced Diagnostics Lab, which is a sort of an innovation space within the Department of Pathology. And this was a seminar that told me about a new, just incredibly innovative and exciting technology that is allowing researchers, and this is a collaboration between researchers at the University of Illinois and our own pathology department, and these folks have built a microscope that uses different wavelengths of light to image completely unstained sections of tissue, either frozen tissue or formalin fixed paraffin embedded tissue, but just unstained sections of tissue. And because of their ability to use multiple different wavelengths of light and different kinds of optics and computational approaches, they can derive information about the chemical and structural nature of the tissue, and then use AI to create virtual stains of almost any type that look completely indistinguishable from the actual chemical stain counterparts. And what's more, these stains actually contain far richer information than the actual chemical counterparts because of the use of all these different wavelengths of light that provide richer set of data. So they call this stainless staining. And coupled with AI, they can construct these algorithms that help us unlock secrets contained within the tissue that we just can't do by eye alone. And again, things I mentioned like prognostic insights, predictive insights into, for example, how a tumor might respond to a particular cancer therapy. To be honest, I don't really understand it all. It's all about AI, and I know that's an incredibly overused term these days. You can't turn around without somebody mentioning something about AI, about literally anything. I almost bought a toothbrush the other day that said it was going to use AI to help me brush my teeth.
Really?
So
Really?
it's like
Oh my
ridiculous.
goodness. Wow.
It's
Well,
all
I mean,
ridiculous.
that's really exciting though. And I agree, artificial intelligence, generative AI, it's going to transform our field. So there is a lot to be excited about. Well, I think I'll just end with my last question in the minute that we have. For the new in practice or the young researchers in the field, what advice would you have for them? The ones that are eager to make a contribution to patient care?
Sure. Yeah. I would say my advice is pretty simple. It's all about attitude. Stay curious,
Mhm.
stay passionate, and I think above all, stay positive and persistent. We know that research can be hard in so many different ways. We're constantly facing rejection as researchers, whether it's in the form of a negative manuscript review or a grant review that you've worked really hard on, that gives you, you get a score that means it's not going to get funded, or whatever. And most experiments, most of the things that you do on a daily basis in the lab, that they don't work, at least the way you expect. But it's those people, the people who succeed are the ones who just remain undaunted in their positivity. I mean, glass half full on steroids, and who just remain persistent at chipping away at their goals, and don't take no for an answer, just keep pushing, because at the end of the day, you're pursuing an incredibly interesting career, and you have a chance, ultimately, to make a difference in people's lives by discovering something new about disease, about its diagnosis, about its treatment. And what better goal can there be than that? So, it's all about attitude, I think, and that
I
persistence
agree
and
with you
positivity.
completely. What wonderful advice. Well, Dr. Lucas, thank you so much for joining us today. I loved listening to your thoughts and plans. I learned about a new toothbrush that maybe I should look into, and I also am just very excited about the field and excited having you here as our new vice chair for research. So thank you again, and we may have you back as a guest in the future to talk about some of the great things you're working on.
Thank you so much for the opportunity, Dr. Pritt. It's really been a pleasure, and thank you for doing what you're doing with this podcast. It's fantastic.
Thank you.
Thank you so much for tuning in to Answers from the Lab. Be sure to subscribe to this podcast, and don't forget to tune in every Thursday and every other Tuesday.